Hello everyone and a very warm welcome to today's webinar on end-to-end solutions for iron-based nanocolloidal products from preclinical development to clinical translation. My name is Surbhi and I represent Veeda Lifesciences. It is a pleasure to have all of you joining us today. Veeda Lifesciences is a global integrated and a technology-driven contract research organization committed to delivering excellence across the drug development lifecycle. From preclinical research and clinical trials to analytical, bioanalytical, DMPK, and life cycles, and regulatory support, we provide comprehensive end to end solutions that help accelerate the development of innovative biosimilars and generic products for clients worldwide. With a very strong global presence and scientific capabilities, Veeda partners with organizations across pharmaceuticals, biopharmaceuticals, specialty therapeutics, medical devices, nutraceuticals, and other life sciences industries. Our focus on scientific excellence, quality regulatory compliance, and innovation enables us to support even the most complex development programs. Today's webinar is focused on the unique scientific and regulatory challenges associated with iron-based nanocolloidal products and explores integrated strategies that support their successful journey from preclinical evaluation to clinical translation. Before we begin, I would like to quickly walk you through the format of today's session. We will start with an expert presentation followed by an interactive Q&A session. You will notice there is an Ask a Question feature available on a webinar screen. Feel free to submit your questions at any time during the presentation. And our speakers will address as many as possible during the discussion. Now, it gives me immense pleasure to introduce our distinguished speakers for today's webinar. Our first speaker is Dr. Vinay Dhiman. He is head at Preclinical at Veeda Lifesciences. Dr. Vinay is an accomplished scientist and research leader with over 17 years of industry experience in bioanalytical sciences, analytical research, DMPK, and pharmaceutical development. He leads Veeda Bioanalytical and Analytical Division, overseeing scientific and operational excellence with ensuring delivery of high quality solutions aligned with global regulatory standards. Prior to joining Veeda, Dr. Dhiman held key scientific roles at organizations including Jubilant Biosys and Syngene International, building extensive expertise in method development and validation, pharmacokinetics, toxicokinetics, and DMPK studies. Joining him today, our next speaker is Dr. Pritesh, Contractor, Principal Scientist, Bioanalytical Research at Veeda Lifesciences. Dr. Pritesh brings more than 19 years of experience in regulated bioanalysis supporting pharmaceutical and biopharmaceutical development programs. At Veeda, he provides scientific leadership in development, validation, and application of LC-MS, MS-based bioanalytical methods in compliance with regulatory standards. He also possesses extensive expertise in elemental analysis using ICP-OES and ICP-MS, with significant experience supporting pharmaceutical products where the analyte originates from the administered drug substance. His work includes analytical and regulated studies involving iron sucrose, iron dextran, ferric carboxymaltose, ferric derisomaltose, zinc sulfate, and several other complex formulations. We are delighted to have both of them with us today to share their valuable scientific insights. So without any further delay, I would now like to invite Dr. Vinay Dhiman and Dr. Pritesh Contractor to begin with today's session.
Thanks, Surbhi, for the brief introduction. And to start with the presentations, next slide, please. Yeah, so iron-based nanoparticles are tiny iron particle dispersed in a liquid. So common examples include iron oxide nanoparticles, supermagnetic iron nanoparticles, which is also known as spions, iron carbohydrate complexes, and iron non-colloidal for imaging and therapy. These formulations have important medical applications such as iron replacement therapy, MRI contrast enhancement, and drug delivery. Also, it is used for cancer hyperthermia treatment. Next slide, please. Coming to the background of the iron-based nanoparticles, these are intravenous iron nanocolloids. These intravenous iron nanocolloids are complex non-biological drugs. These consist of an iron oxide core surrounded by a carbohydrate coating that stabilize the particle. Example include iron sucrose, ferric carboxymaltose, sodium ferric gluconate, and iron isomaltoside. Because of their complex structure, two products may look chemically similar, but still behave differently in the body. Simply proving that the two generic formulations may look same under the microscope or have the same chemical formula, it isn't enough for regulatory bodies to consider them truly identical or interchangeable. This creates a unique hurdle for standardization or replicating these products. Next slide, please. Now, coming to the EMA reflection paper with which we are working on these biodistribution studies. So, why are these additional preclinical studies required in first place? It comes down to an inherent nature of these nanocolloidal iron products as they exhibit a highly complex structure, critical quality attributes that are different to fully characterized in a standard lab settings, and consequently potential difference in their biological fate once inside the body. Therefore, physiochemical similarity alone is not enough. Hence, we need preclinical biodistribution studies to compare the tissue distribution, organ uptake, iron release, clearance, and retention. The goal is to confirm similar in vivo behavior between the test and product. Next slide, please. Primary objective is to directly compare the biodistribution profile of the formulations, to evaluate organ-specific iron uptake, and to assess RES accumulation, where RES is the reticuloendothelial system. Basically, we look closely to how the reticuloendothelial system, like the liver, spleen, handles and store these nanoparticles. The secondary object is to gain the long-term cell safety, to compare the elimination kinetics to see how quickly the body clears the iron, and to assess long-time retention to ensure no toxic buildup occurs over time. Importantly, we actively scan to detect any unexpected tissue accumulation in non-target organs. Rodents are the preferred models because their iron metabolism is well understood and test and reference products are studied in parallel. They are incredibly sensitive for tissue distribution assessment. There is a vast established database on rodent iron metabolism. Finally, the underlying studies rely on parallel administration layout. This means the test product and reference product are evaluated side by side in separate synchronized groups under identical conditions to ensure any observed variation is purely down to the product characteristics themselves. Next slide, please. So coming to non-clinical rationale, EMA expect biodistribution studies to compare plasma and RES distributions, tissue accumulation, iron release, and clearance. The data must come from a validated analytical methods, GLP compliant studies, and appropriate statistical analysis. Next slide, please. The mechanism of RES recognition. The primary recognition pathway relies on RES macrophage phagocytosis. When these particles enter the bloodstream, they are highly cleared by specialized macrophages in the specific organs. Mainly the liver, specifically handle the Kupffer cells. The spleen, beyond standard macrophage engulfment, we also observed endothelial and epithelial uptake. This includes hepatocyte endocytosis, direct endothelial uptake, along with the vessel's wall. Crucially, this behavior has a high surface chemistry dependence. The exact coating of the nanoparticle dictates how heavily these cells interact with it. Next slide, please. Coming to interspecies variability, while a relevant model is required, when evaluating complex nanocolloidal iron products, we face serious translation limitation if we rely solely on basic surrogate or in vitro models. Animal models are essential because they provide information then that cannot be obtained from cell culture or blood samples alone. First, while animal and cell models can capture some PK aspects or pharmacokinetic behavior, they don't tell the full story because of this high complexity. High human blood and plasma data alone cannot adequately describe distribution. Just looking at how long the product circulates in blood doesn't tell us where it actually deposits in tissue. Therefore, we arrive at our core operational conclusion. In vivo study in a relevant animal model are absolutely essential. We cannot skip this step because we need to complete living system to fully capture the and cover the distribution, metabolism, excretion, and degradation products of the nanoparticles. To this ensure we are tracking not just original compound, but exactly how it breaks down and leaves the body. Next slide, please. Coming to target organ selection and dose finding, there are two primary required compartments we must monitor. First is the vascular compartment, which includes plasma and RBCs. Second is RES, where the spleen is highly recommended for tracking iron accumulation and storage. When we look at the sampling time point themselves, the strategy is meticulously structured. We focus heavily on early time points under 24 hours to 72 hours, which is critically for capturing the active windows of RES clearance. The overall timeline must cover the full concentration time profile to accurately map the kinetics. It must dynamically reflect both incoming iron and its subsequent tissue release. Finally, we conclude an extended time point for tissues to measure the long-term accumulation and delayed clearance. And the selection logic behind this is to ensure that all organs identified in the reference drug profile are actively tracked. Cross-reference and confirm those same organs are identified in our test group also. Confirming that all three core compartments are completely covered. And finally, the strategy is optimized to achieve true cellular level resolution, allowing us to see precisely how the nanoparticle distribute down to the microscopic cell level. Next slide, please. According to the iron nanoparticle disposition, the liver, spleen, and bone marrow are primary organs of interest because they clear most nanoparticles. Additionally, tissues such as kidney, lung, heart, and lymph node are also examined to detect unexpected accumulation. And to go along with, if we are giving a dose of a known level, and we can see what would be the percentage of the injected dose present in different part of the tissues. Next slide, please. Now coming to the core competency of method development and analysis, we have successfully developed the methods in plasma, serum, as well as in different tissues using ICP-MS. Iron levels are measured using the validated analytical techniques such as ICP-MS and ICP-AES. Histological staining and microscopic complement these measurements by showing where iron is located within the tissues. So we also suggest to have some histological staining and microscopic complement assay for checking the amount of iron in the tissues. Our basic method requirement dictate that we must have fully validated quantitation methods in blood and plasma, fully validated quantitation method across our target tissue. As we execute this, we keep three critical concentration in mind. We must critically control sample processing to avoid sample degradation or contamination, and constantly verify the suitability of bioanalytical results. Also, we must thoroughly document data in interpretability per matrix and recognizing the blood behaves differently than solid organ tissues in an assay. For plasma, ensure there is a clearly stated LLOQ and finely documented recovery rates. For our target tissue, we establish a tissue-specific LLOQ for which unique organ and utilize a matrix-matched calibration strategy. This ensures that the dense tissue of a liver sample won't artificially skew the reading compared to the spleen sample. So, coming to method of analysis and data interpretation, do we need to separate free iron from nanoparticle bound iron in our tissue assays? As it turned out, measuring total iron suffice for our study objectives. There are four key reasons for this. First, the coated particle degrade gradually within the body. This means time-dependent release directly reflects the degradation process itself. Furthermore, the intravenous iron loading is incredibly large compared to the baseline. Physiologically, iron levels already present in the rodent model. So data presentation framework, we proceed like when we compile this data for our regulatory review, the data presentation follows four strict metrics to ensure the transparency. We report iron per gram for the tissue, percentage of administered dose, mass balance recovery, and time course per compartment. To establish definitive evidence of for similarity between our test product and the reference standard, we look comparable findings across our four pillars. A similar organ distribution profile, similar uptake kinetics, similar tissue clearance, and a similar retention profile. The potential concerns with this would be excessive liver accumulation, which could signal poor surface coating stabilization, increased splenic sequestration, sorry, sequestration, indicating over recognizing by macrophages, delayed clearance, which raises long-term toxicity and retention concern, or the appearance of completely new target organs that the reference drug never accumulated in. Next slide, please. Coming to the estimation of total iron and transparent bound iron, these are the LLOQ which have been established at our premises with using the ICP-MS as an analytical tool. We also perform the serum analysis in for transparent bound iron. Next slide, please. Coming to statistical approaches, because we are dealing with a complex biological system and multiple tissue matrices, our approach must be both robust and proactive. We start by acknowledging our data complexity. We are managing longitudinal data across many endpoints, meaning we track changes over multiple time intervals. This spans multiple compartment and tissues as we saw in the earlier slides. Quantitative analysis of sparse data commonly challenged in preclinical tissue studies are the quantitative analysis of sparse data because animal must be sampled destructively at individual time point and we cannot track the single subject through an entire timeline. To overcome this, we utilize a dual modeling strategy. First, apply standard non-compartmental analysis to calculate three critical pharmacokinetical variables, that is Cmax, Tmax, AUC. And secondly, employ PBPK empirical modeling, which supplements NCA data with PBPK. This mathematical modeling allows us to specifically account for destructive sparse sampling and reconstruct a continuous kinetic profile. Ultimately, report summary parameters for both analytes side by side to give a complete transparent picture of biosimilarity. Next slide, please. So, Coming to the study design can be sponsor specific, or we also make a study design and give it for the sponsor for their review. And looking at the metrics on this left, we break down our fundamental study parameters, that is species. We utilize rat model aligning with the rodent preference as justified earlier. Groups, the animal cohorts are divided explicitly into parallel test and reference groups. Route of administration, strictly it is IV. Those both groups receive the equivalent iron dose to ensure true head-to-head evaluations. Coming to the time point sampling is highly dynamic, starting immediately at 1 hour, followed by 24, 7 days, 14 days, and 28 days, an extended window of 6 to 8 weeks to monitor long-term behavior. This way will perform for the tissues. However, for the plasma and serum, the time points will be from 1 hour till 72 hours. Coming to organs, major organs are liver, spleen, kidney, and bone marrow. And coming down to the biodistribution endpoint, we monitor blood compartment. This requires measuring of total iron concentration and tracking the continuous plasma kinetics profile. Second analysis will be tissues, where we measure the absolute iron concentration per gram of the tissues and map the overall distribution pattern across the exact organs. Third, we will closely follow the rate of tissue elimination and calculate the total physiological recovery over time to prove the body is processing the iron successfully. Finally, this focuses heavily on the persistence after administration, utilizing our extended 6 to 8 week time point to verify that the test product does not over retain or lock itself into tissues compared to the reference standard. Next slide, please. Now, coming to the regulatory expectation, the EMA expects no clinically meaningful difference between the test and reference products in distribution, accumulation, iron release, and elimination. Meeting these benchmarks builds what regulatory call a supporting totality of evidence. This means our submission package must stand on a three prolonged foundation: robust quality data from manufacturing comprehensive, non-clinical data from our living models, and highly detailed PK and distribution data, specifically defining acceptable similarity margins. Because these are non-biological complex drugs, rather than simple small molecule, establishing the exact mathematical boundary for what qualifies as similar remains an incre- inter- sorry, intricate process that requires close collaboration with regulatory agencies. Next slide, please. Now, coming to the conclusion and key takeaway from our evaluation, we must reiterate that iron nanocolloids are complex medicinal products, making them distinct from traditional small molecule drugs. This is because a product organ distribution directly impacts both its efficacy and its clinical safety profile. Utilizing advanced analytical tools such as ICP-MS framework, as we discussed, enables precise tracking across highly diverse biological matrices. Deploying integrated end-to-end solution escalates from preclinical to clinical translation, taking us smoothly from preclinical animal models into clinical readiness. Ultimately, generating robust distribution data strengthens our regulatory success and minimizing submission risk. Next slide, please. Now, coming to end-to-end workflow, I have made this workflow in a chart. So this is how we do a complete development pathway as well as the experimental flow, how we perform this. So nanoparticle administration, delivering our parallel test and reference doses intravenously. Then the blood collection at our specified early time at our specified early time points, organ harvesting on our target tissue sets, rigorous sampling preparation to prevent contamination or degradation, validated assay for iron measurement by ICP-MS, An instrument readout undergo through data analysis, and finally, we apply mathematical framework to generate the PK biodistribution modeling. This process generates the evidence needed to compare the test and the reference product. Thank you, and it's over to Dr. Pritesh, Contractor.
Yeah, thank you, Dr. Vinay, for such nice presentations and insight explanations. And thank you, Surbi, for the introductions during the beginning of the sessions. So after the preclinical discussions, we can move and discuss for the BAB analysis and how the iron formulations and specifically the FCM can help us, and how we can do the method development validations and its implements to the study sample analysis, so that we can discuss in today's sessions, and then we can also discuss any queries and questions during the Q&A sessions. So let's begin. Please do the can you please next slide. Fine. We can start with some background of these iron formulations. So, why the iron formulations as IV is required? And of course, in the market, there is the oral formulations are already available. Then why should we develop the IV formulations for our these studies and method development and validation? So what happened? The many times the oral solutions are not effectively work for the patient. Actually, they cannot absorb in the GI tract, or maybe some of the patients may not tolerate the absorptions or of the iron from the oral formulations. So the IV formulations can be worked, and it is directly given bypassing the GI tract, and that help us to the patient pool who really requires the iron when they have suffering from the iron deficiency, specifically for the patients who has suffering from the anemia, patients who have the chronic kidney disease, and the pregnant women who may require the iron during their pregnancy periods. So these are the patients who they definitely required iron preparations, and to maintain their normal iron level in the body for the day-to-day life. So now why we have to do the developments, and what the regulatory says for the this development validations and study, and how they are take up and suggesting to do the activities. So here I have presented the regulatory guidance documents. There is a two approach for the measurement of the iron. One is the ICP-OES assay. Second one is the size exclusion chromatography by using the ICP-MS techniques. And bioanalytical methodology, what we are going to use, the reference and working standards, what we are going to use during the development and validations, as well as the study also. Then challenges and the resolutions, what we have faced during the development sections. Then a method validation summary, how our method validation summary can help our clients for the patient-based study, because generally the patient-based studies are going to a lengthy process and it's continue for 2 to 3 months. And the Veda's experience and the expertise, so that we can show you our progress and our progress card in front of you, how expertly we are handling the client study, right? So please move to the next slide. First, we start with this guidance document. This is the OGD from the US FDA, revised and released in the November. And if you see here, they are introduced two different options. Option one, they are newly introduced. This one is not earlier available, but for this formulation, this is newly introduced. They recommended for the measurement of the ferric carboxymaltose associated iron in serum sample. And the second one is the option B, where they recommended for the two separate analyte. One is the total iron in serum, and second one is in the transferrin bound iron in the serum. And they also provide the how to measure on which PK parameters to be taken care for the evaluation of the study outcome. Okay, so the option A is the newly introduced, definitely that we have to discuss in our presentations, and we also show you our data, what we have at present. But we can start with the option B first because it is a normal analysis and it is available and accepted by widely by other industry colleagues also. So let's first discuss with option B and then option B. Please next slide. Okay, so this option B specifically, we have developed and validated the method by using the ICP OES equipment, which is the optical emission spectroscopic based method. Next slide, please. So here two separate method is required. The first method is the total iron estimation in the human serum samples, and the second one is the transferrin bound iron in a human serum sample. So the total iron method is quite reasonable and it's easy to develop and validate because it is giving a completely total count of iron present in the serum samples, and that can be there is some easy steps to be followed, and then we get the estimation of the total iron from the system. But the challenging part is the transferrin bound iron, because transferrin is one of the protein which is going to be used for the circulation of the iron in the human body, because it is transporting the iron after the administration of the IV formulation, and we have to isolate this TBI from the other unwanted iron species like some free iron when the iron is administered. Otherwise, in healthy volunteers or in healthy subjects, the free iron content is not present. But when it is administered, there is less quantity of free iron may possible, so that we have to isolate. Then drug bound iron, that also we have to remove from this measurement of TBI, so that we can see in the next slide. Next slide, please. Okay, this is some basic parameters of our equipment that we have used, optimized during our method development sections. So this is the technique that I have explained. It is the OES techniques, the inductively coupled plasma optical emission spectroscopy. The equipment that we have used is the Perkin Armor Optima 8000 series, as well as we have a new version of equipment is also available with us. The auto sampler details are giving, the software version is also giving. Our software is also we have connected with our LIMS database. So our final data we have to generate from the LIMS. So that is one of the beneficial point for us also, as well as for our client also. So the final output of all the data for other generic drugs or LCM, SMS analysis or ICP-OES, so those all are the same in a template or in a presentation point of view. And it's also easy for the auditors and reviewers to review the data which is generated from the LIMS database. Then the wavelength related details are mentioned. Some ICP OES parameters we have mentioned in these slides that we have used during the development sections. The ISTD details we have mentioned in these slides. The carboplatin we have using as ISTD. Intensity and the linear equation is the y is equal to mx plus c. The regression model is the 1 by c square, or we can say the 1 by x square, that we have used during the analysis. Okay. Next slide, please. Now, these are the related to the working or reference standards. So the total irons, we are directly using the formulations for the preparation of our stocks, calibration curve standards and the quality control samples, and that can be changed from the formulation to formulation. So if we are working on the FCM, then we can use the FCM formulations. If I am working for the FDM, ferric derisomatoes, so I'm using that formulations. If I am using for the iron sucrose, so I can also use that one. So it's changed from formulation to formulations. For transferrin bound iron, we are using the holotransferrin, so that is easily available from the commercial vendor. And that's the Sigma Aldrich, and some few or others are also available. Then ISTD, as I have already explained, it's a carboplatin that we are using, and that is also available from the commercial vendors. Next slide, please. Okay, here the linearity range we have mentioned in these slides for the sample processing volume. For the total iron, it's a 0.1 ml, and for TBI it is a 0.75, means only 75 microliter we are using for the TBI. And the LLOQ, you can see it's a 0.4 to 800 microgram for the total iron. And for TBI, LLOQ remains same, 0.4, and the ULOQ is 16 microgram. So this range is quite decided and set by keeping the endogenous level of the serum in mind also, as well as we have taking the data from the healthy volunteers as well as the patient pool, and accordingly the LLOQ has been set. And these are the remaining QC concentrations presented in these slides that we have used. Now the ULOQ is the 800 microgram per ml we have take up for the total iron, because when the patient based study is executed, so the dosing is about 1000 mg, so 1 gram somewhere. So when the 1000 mg is dosed to the patient, so the anticipated Cmax value for the patient is about 450 to 600 microgram somewhere it was observed. So considering that point we have keeping our ULOQ at 800 microgram for the FCM formulations. Next slide, please. Okay, think this is one of the limitations for this method that what we have developed. It is when the samples are hemolyzed. Okay. So what happens, this hemolyzed nature of the sample is going to interfere with the analysis of the iron, because the hemolysis nature of the sample itself, it's due to the iron, it's ruptured of the cells and everything, the blood cells. So that is going some spike during the analysis. So this is one of the limitations that we have observed during the analysis also in development section. So the hemolysis samples are identified, recorded, and we are not going to do the analysis of the hemolysis samples for this iron analysis. So this is one of the limitations. The scale chart is provided to our phlebos and other CRS persons for the identifications of the hemolysis samples for this iron study. Next slide, please. Okay, now this is the stabilities what we have proven during our method validation sections for the total iron as well as for the TBI also. Here we have provided the stability of extract. It is 69 hours for ambient and 77 hours for TBI. The freeze thaw cycles are also presented in this slide. It's a 5 cycle, -20 and -78. Benchtop stability at ambient temperature, 76 hours, 73 hours. Auto sample reinjection, 47 hours, 27 hours. Long-term stability, LT stability in matrix for total iron and TBI. It's 339 days for TOI and 338 days for TBI. So this part I am talking about earlier, because what happened when we are talking about the patient-based study, so it is going to be for different site and subjects and the patient's recruitment and everything is going on. So the clinical duration is about 2 to 3 months duration for the patient-based study, and for the bioanalytical we may require around 1.5 months or 2 months for the analysis. So considering 1 to 6 month durations, that entire duration we have to cover during the LT stability also. So here we have established the LT stability for the 339 and 338 days. So we can do the analysis within the stability period for the patient-based trial. So there will be no any hiccups for the LT stability, and there will be no any requirement to initiate the analysis in between group-wise, because we have a sufficient stability duration available. The remaining is the system-related stabilities, the batch size stability, the dilution stability, that is as per the routine regulatory guidance requirement, ICH M10 or FDS and other regulatory guidance requirement. So those all stabilities are available for this validation. Next slide, please. This is one of the important experiment that I have to explain. This is the extraction cartridge efficiency experiment specifically performed for the transferrin bound iron method validation, because in the beginning I have explained that why it is important to verify and assure that actually we are measuring the transferring bound iron, which is not going to be interfere with any unwanted iron species. So that's why we have introduced this experiment. We did the stability also, the benchtop stability, the freeze-thaw stability, to check those effects, as well as the LT stability. What in this experiment, what we are doing, we are separately prepared the QC samples. Those QCs are separately spiked with the formulations with the concentration of 600 microgram per ml, because earlier I also explained that it is a Cmax of the total iron. So we selected the Cmax concentration of the total iron. By keeping that things in mind, we are preparing the formulation spike QCs for TBI, and then we can do the analysis and we check the efficiency of the extractions, that any unwanted iron should not be eluted along with the TBI. So the TBI level is going to be properly measured, and there will be no any false measurement of the TBI by adding any unwanted iron. So this experiment we did, we validate during the validation. And in addition to this formulation spiked QCs, we are also using during our study sample analysis when we are doing the TBI analysis. So that is the additional checkpoint for us also. So we are assuring during each and every analytical run for the TBI measurement that the QCs should be found well within acceptance criteria, which provide assurance that the measurement of TBI is completely accurate. So this is one of the good things that we are doing for our client. We provide the more robust and reproducible data to our client. So the regulatory will not raise any questions or query related to the accurate measurement of the TBI. So this is one of the best things for our method. Next slide, please. Okay, so the till previous slide, I have explained everything about our option B. Now in this option A, this is the newly introduced. What happens in this option A? In option A, the measurement what we are doing, it is a ferric carboxy maltose associated iron. So here no the total iron is in picture. There is no any TBI is in picture. It is completely a different species we are going to measure, which is the formulations which is connected or associated with the iron. So that only part we are going to measure, and by using this LC-ICP-MS method. So how this new technique is going to be used, either we can say this is a hybrid technique we have developed and validated, so that we can see in the next slide. Please, next slide. Okay, so in this study, what we are using, the size exclusion chromatography we are using, and that is coupled with the ICP-MS. So the size exclusion chromatography gives a proper separations of different iron species which is required. From that, we are picking the formulations bound iron, either we can say a ferric carboxy maltose associated iron, and that we can measure by using this size exclusion chromatography. Next slide, please. Okay, so by using these techniques, we are separating this associated iron and then transferring bound iron, as well as our IS3D also. The linearity range we have set for this measurement is 600 microgram to 10 microgram. Here you can see the LLOQ is the 10 microgram, because why this associated iron is not endogenously present. Okay, we are talking here about the associated iron. We are not going to measure iron which is endogenously present. So therefore, considering the anticipated Cmax values of this associated iron, the LLOQ is keeping like 10 microgram iron in the beginning of this development. Overall runtime of this method is about 20-minute runtime, plus another 2 minutes is for the rinsing cycle to remove the carryover from the system. Right. So these types of method we have set and developed for this FAI measurement. Next slide. Here you can see the representation of the chromatogram. This is from the size exclusion chromatography ICP-MS. Here you can see the 600 microgram per ml FAI. The first part of this figure is the ULOQ, The bottom part of this figure is our LLOQ, and the peak which is showing at the right side, it is the ISTD peak for the carboplatin that we have used for this measurement. So these types of chromatography we have getting in our ICP-MS system for this FA analysis. Next. Now, this is one of the good slide for our client, for our sponsor, and for our industry colleagues who joined at this moment. You just see how we can ensure that our FAI method is similarly working with our TOI and TBI method. This is one of the exploratory study we did for one of our client, and you see here the data. The third column, you can see the total iron concentration marked as A. So that data is measured and run from the ICP-OES equipment. The TBI, this is fourth column. The concentrations derived from the ICP-OES analysis, and the FAI, here it is mentioned like a C. It is a measured from the size exclusion chromatography ICP-MS method, and this is the concentrations practically what we observed during the analyzing these samples on the SEC LC ICP-MS. And the FAI, the D column, is the theoretical value when we are subtracting TOI minus TBI, and what values we are getting, that is also mentioned. And the last column is the percent difference between the theoretical value and the practical value, and you see the how the results are impressive. So this gives us the confidence that our method is perfectly working. It is a reproducible, robust, even though we are using the different equipment, even though we are using a different analytical technique. But finally, what we are going to target to measure, that we are getting with this new approach and the new techniques. So with this, we are providing a better solution to our client and our sponsor as per their study requirement. Next slide, please. In beginning, I have just explained about our card, the progress card expertise. You see the list of studies we did for TOI, TBI by using the ICP OES. Here is the ISR acceptance we have mentioned, and this is all the good and accepted as per the ICH M10 and the regulatory requirements. So this is the output of our expertise, our methods, and for our clients. So this is the things what we have to explain in this webinar to all our colleagues and the sponsors, that how confident we are for your programs. So we are encouraging all of you. So if you have any queries and future feasibilities for executions of the iron preparations related studies for BAB healthy volunteers, or maybe for the patient-based studies, so please reach out to us so we can give you the best solutions. Next slide, please. Fine. So this is the explanations from my side for the BA part. So we can start question answers. So we can take over first.
Thank you, Dr. Pritesh and Dr. Vinay, for that insightful and informative presentation. We will now move into our interactive Q&A session, where we will address some of the questions that has been submitted by our audience throughout the webinar. So the first question has come for Dr. Vinay. And the question is, why rat over mouse as the default, and when would a non-rodent be justified given interspecies observation differences?
Yeah, thanks, Ubi. So the rat is generally preferred over the mouse as the default rodent species for non-clinical pharmacology and toxicology, because it provides more reliable characterization of the drugs, macrophage-mediated disposition, hematologic responses, and safety profile. A non-rodent species, example dog and mini pig, is justified only when there is a evidence that the rodent model is not sufficiently predictive, particularly because of the species difference in opsonization and RES something.
Okay. Next question is for Dr. Pritesh. So the question is, what are the challenges have you faced in developing ferric carboxymaltose associated iron methods?
Okay, it's a good questions, and it is really very learning for us also, because we have also doing this development first time, because the size exclusion chromatography associated with the LCMS for the BAB study, certain setup we did first time. So first of all, the selection of the analytical column is one of the challenge. How to select the size exclusion column, which parameter to be taken care, so that we have to study, and accordingly we have selected the size exclusion column. Then better mobile phase, a composition of mobile phase that we have to select, who supports and suitable for this SEC column, as well as the ICP-MS analysis also. So that we have also evaluated, we have checked some compositions and everything, and then with that we have fixed the mobile phase for the better separations and the resolutions of the different species of the iron. That was one of the challenge we have observed during the development phase. Second and the third challenge is the carryover generated from the systems, because as I have said, the ULOQ concentration is very high. It's about 600 microgram per ml. So when the such high concentration are running from the systems, it gives some residual effect for in the next sample. So that we have to completely wash out the system tubings and the system line properly between the two runs. So that carryover issues we are observed, so that we have to resolve by using the bio inert LC that we have specifically procured for this analysis. And with the combination of some rinsing solution, strong and weak, we used it, and accordingly we can resolve that carryover issues. So definitely those some of the challenges we observe, which is not routinely observed in our day-to-day LC-MS analysis, but when we talks about size exclusion chromatography, it observed. But scientifically we take up this one and resolve and solve our all challenges. Thank you.
Thank you, Dr. Pritesh. Next question is again for you, and that is why do different equipment ICP OES and SEC ICP MS for different way of measurement of formulation bond iron, total iron, and transferring bound iron?
Okay, as during my discussion session, I explained that the FDA suggested two different approaches, option A and option B. So definitely our client has some requirement. Maybe some of the clients may require the option A, some of the clients may require the option B. So till date, a numerous study we completed by using the option B, total iron and transferrin bound iron measurement, and on ICP OES equipment. We submitted those studies in Europe as well as in the US FDA also. So it is accepted very well. So that confidence we have, and the FAI is a different approach. Some of the clients are really asking for that, because in that only the one analyte is there. Of course, some financial implications are there, because the client's requirements are very as per the budgets and everything. So from as a CRO point of view, as a service provider, we are ready with both the options to meet our clients' requirement. So therefore, we have all methods validated and available to meet the client's program of this iron preparations requirement.
Okay. Thank you so much. Next question is for Dr. Vinay. What is the realistic timeline and budget for a GLP compliant comparative biodistribution program?
Yeah, so for GLP compliant comparative biodistribution program, the timeline is generally 6 to 8 months, including method development, validations, dosing, sample collection, and sample analysis. And for the budget, it will be approximately around 5 lakh USD.
Okay. Next question is again for Dr. Vinay. How does ICP-MS distinguish injected nanoparticle iron from the body's large endogenous iron pool, especially in liver and spleen?
ICP-MS cannot directly distinguish injected iron carboxymaltose from the body. Endogenous iron, because both contain the same naturally occurring iron isotopes. So conventionally, ICP-MS measure only the total iron concentration in the tissue such as liver and spleen. In most preclinical studies, the contribution of administered FCM is estimated by comparing tissue iron level in treated animals with untreated controls and subtracting the baseline endogenous iron content.
Okay. Next question is for Dr. Pritesh. Being an endogenous presence of iron in human serum matrix, how it is accounted for accurate measurement of calibration curve standards and equality control samples as well as for study samples too?
Fine. It's really very good scientific questions. As we know, the iron is definitely present in the human body. So how we have to account for the true measurement of the iron which is administered from the IV formulation. So for calibration curve and for the quality control samples, we are using a blank subtraction method for regressions of the iron formula, iron CCs and QCs, and that's why we are using our Watson LIMS database for the final regressions and the results preparations. So whatever the endogenous concentrations presents in that particular serum lot, that endogenous concentration is first of all going to measured and subtracted from the calibration curve standard. And as per the ICH-M10 guidance document requirement, we are using zero EC samples for quality control samples measurement also, and we are going to do the amendment of this endogenous level to our nominal concentrations of QC, and accordingly the calibration curve standards and QCs are finally calculated for the analytical run acceptance. And when we talks about the study sample, so for study samples, pre-dose samples are definitely collected. Either it is for a Europe submission or either it is for a US FDA submission. So for both the regulatory requirement, they have defined the parameters for the PK study evaluations. And based on that, baseline corrected data has been generated. Whatever the concentrations or whatever the area observed in the pre-dose sample of particular subject or participants, that should be subtracted throughout the post-dose samples, and accordingly the PK study evaluations has been done. So with this combined approach, we are taken care of this endogenous level of iron.
Thank you so much, Dr. Pritesh and Dr. Vinay, for sharing your expertise and for addressing so many insightful questions from our audience. We truly appreciate the enthusiastic participation from everyone who joined us today. Due to the time limitation, we may not be able to answer every question during this live session. However, please be assured that all unanswered questions will be responded via email after the webinar. We will also be sharing the recording of today's webinar with all registered participants so you can revisit the discussion or share it with your colleagues. On behalf of everyone at Veeda Lifesciences, I would like to sincerely thank you all for taking the time to join us today. We hope this session has provided valuable scientific insights into the analytical, bioanalytical, translational, and regulatory considerations involved in developing iron-based nano colloidal particle products. A special thank you once again to our speakers, Dr. Vinay Dhiman and Dr. Pritesh Contractor, for delivering such an engaging and informative session. We look forward to welcoming you again at our future webinars. Until then, thank you, stay safe, and have a wonderful day.
Thank you so much, everyone. Thank you so much, Surbi.
Thank you.
Thank you.